After you completed editing the file in Photoshop, I'm going to show you how to organize those files into a folder structure that you can easily use in programmable airspace profiling software. Here I'm just going to show you with one image. First, your base folder structure should be like the project name, assuming this is an animal study, some with smoke exposure, some with just the room air. So this layer we're gonna name them smoke versus air group. Oh no, smoke versus air project. Let's just call it this way. And move this file into it. Of course, in real experiment you will have a lot of these files. And the next layer is recommended to create for different groups. So first group, let's just call it smoke group. Second group, call it air group. Okay, this one, it's rather healthy. It's coming from the air group. So we're gonna put them into the air folder. And of course, you have many biological replicates. So in this case, let's assume there is one animal, one individual, that's called A01. Then you should put this file into that one. And at this level, you can decide to either leave it in here or create even more detailed folders, such as section 1 or section 2, but it's up to you. I think it's perfectly okay if you have one layer of group, another layer of each individual. So let's just leave it this way. Okay, after you organize the folder structure, let's launch the software. At the time of the recording, we are using the November 2016 version. Okay, we can maximize this, and just to show you a few things. Among settings, if you're not sure about which setting to use, actually you can always start with the preset test large. When you are using large images, this is the way to load all the presets for large images. And layout, you can play with this as well. Show all panels, change to this. Okay, and tabbed panels. Yeah, these are two different style of layout that I really liked, with courtesy to the Avalon doc. Very helpful .NET framework that I can use to build this nice looking graphical user interface. And theme, there are a number of themes here. I'm okay with the arrow. You can experiment with the rest of that. Okay, after you use default test large image, it's actually pointing towards test large folder because I included one test image there. But for your own experiment, you need to point that to where you have your projects ready, so which is here. Do not point deeper into the folder structure. It's okay, just click the project. And next layer is going to be the group, and next will be individual underneath that. Okay, just point here. Okay, click OK. Usually it automatically scans for all the images. We only have one. It clearly recognized the group, the first layer, the second layer, and file type, width, and height. You can check these to apply filters if you have too many images. Okay, so whenever you single click, it will show you all the views. Let's just go back to show all panels. Okay, view 1, 2, 3, 4, 5, 6. Okay, you can view them directly here, or you can use a scroll viewer to view each individual one of the image. Okay, well, the first one, view 1, is going to show you the original image without anything done to it. The second one is going to show you markup, applying some of the different things that if you check white balance, Gaussian blur, these will all affect the second image. Okay, and the third one is excluded, which area is going to be registered to be excluded. So in this case, since we already have them painted green and check this exclude color green and color radius, clearly it's recognizing correctly. All the green area has been marked for exclusion. And next is gonna show you the gray and fifth is going to be pure black and white. The sixth is going to be the result. So the helpful thing about the scroll viewer is that you can just drag it along, okay, and have any size you want, and check whichever image you are targeting, and then zoom in and out to do this, and also pan around very easily. Okay, right. Yeah, and whenever you switch between different view, it's not going to shift place. So the green color, as soon as I click excluded, they are matching exactly. And there's the shortcut key. You can press Control 1 to go to the first view, Control 2, 3. Yeah, you can use this shortcut key to quickly switch between them. Okay, let's look at the settings. First, the project name. This project name is sometimes you have all the images, but you want to perform the analysis with different settings on different date. So whenever you do that, you want a separate resulting folder to save. So later you do not find your results overriding each other. So finally, you have several folders of different sets of runs that you can choose from, okay, and compare their results. So this is the purpose of having a project name. Pixel scale is extremely important to calculate the area values or the lens related values. It's how much micrometer each pixel occupies. So in this case, we generally use 10x objectives, so it's equivalent to about 1 micrometer per pixel. But check with your microscope or your software to make sure you enter the right number. Resize ratio, generally the ideal number would be 1, which means there is no resizing. All the analysis will be done at the original resolution. But if your computer is a little bit slower or your pixel scale here is small enough, usually the more detail it has, the smaller the pixel scale will be. So you can actually set this a little bit lower than 1, but don't set it too low that if you do not have any detail in the image, then your result will not be good at all. Okay, white balance. If your white area is having some yellow or some weird tint, you probably want to turn this on and see whether it improves. If it improves, you can see it from view 2. But in this case, I think it's fine. Gaussian blur, if you check this, it will blur the image a little bit, but it will significantly slow down the process, and most of the time it's not very necessary, so you can experiment with this. I generally do not turn it on. Exclude area, it's extremely important to exclude those non-parenchymal areas, so always check this and set the right color and confirm with view 3 whether it's recognized correctly or not. And down below we have binarization settings. Generally for the whole slide scan, if you see an image like this, all the corners, the inside, everywhere is having a very even brightness, then I recommend using global threshold and using an absolute value. So anything brighter than this value will be considered white. Darker, lower than this value will be considered black. So it's some long structure. So generally use this for the large scan. If you have a small view of the scan by taking with a microscope, whereas the side where you can see some light fall off on the corners, in that case you may want to switch to local thresholding. In that case, it will have a moving window with this size and compare the brightness difference. It's going to help a lot if you have light fall off, like a vignetting effect. But for this scan, always go with global thresholding. There are a few settings here that I can explain a little more. These are binarization settings, okay, and also has to do with parenchymal space profiling, of course. This first one, fill holes in airspace and fill holes in tissue. Let's just run an easy example. First, just to run a thresholding on this image. When these two settings are checked and with a certain number in it, it's going to find out certain area. For example, this one. I did not clean it very well, but this is clearly a hole or a blemish within a large air. So when you check this on, this area will be excluded. Fill holes in tissue will do the opposite thing. Sometimes, and quite often, you can see some macrophages in your lung tissue. So in that case, if you check the fill holes in tissue and fill holes in air, not only you get rid of all the unexpected small blemishes off, and you can also clean some of the macrophages. Okay, it looks like I just clicked the threshold, and the threshold result is here. Also, view 5. Let's look at this area. When you switch between view 2 and view 5, you can see here, this macrophage is gone. It's no longer there. Okay, the other area I mentioned a little earlier, this piece, it's not there, right? But what happens if you uncheck these two and run the thresholding again? Okay, you will see the result in a few seconds. Okay, next, morphological dilation, morphological erosion. I don't usually recommend checking both of them and set 1 to each of them. Generally, doing both dilation and erosion will connect to them, whereas but still not moving the boundary in one direction too much. So I generally recommend checking both with 1. You can slightly increase this, but don't set a too large number. And you can, of course, uncheck both of them to just not apply these operations. Okay, here are the results it's in. This area, without checking that, is showing. Okay, and macrophages everywhere, it's showing, right? Like this one, and that, I'm pretty sure. So if you want to get rid of those, make sure you check on these two settings. That's what those are for. Okay, and minimum size. This only applies to the next step. After you perform the basic airspace profiling, there will be so many airspaces out there. So this minimum size will tell the software any airspace smaller than this are generally not correct size, are like too small or like noise in the image. Basically, you can just look at an area that you generally consider a single alveolus size. If you generally set this minimum size, check it and make it small enough that even a single alveolus is much bigger than this. It will get rid of a lot of noise on the low end. Maximum size set to 6 is going to make sure that all the area outside of the lung, interconnected, the huge airspace, will be excluded, not be considered a small airspace. So that's why these two are important, and you need to always check them. Okay, so this one shows they did some profiling and calculated the two cutoffs. Okay, let's just see how the cutoffs looks. So now, after doing the first step profiling, it's going to tell you like how each airspace has been recognized. Okay, looks like there's some problems, like this area. This area is actually black, so it's blended. Somewhere the airspace is broken and it blended within the rest of the area that's outside of the lung. And also, this area clearly should not be all connected. Each airspace has been assigned a random number, but if such a large area share the same color, you start to wonder whether they've been considered one large airspace. It's clearly wrong, right? They should be divided into smaller areas because it didn't pick up a strong staining and too bright here, so it's been mistakenly characterized as one large airspace that connected together. Okay, and their threshold method, all of these are connected and marked as red. So there's the problem. Also, as I mentioned in my paper, the blue line in a normal condition, in a normal developed mouse lung, the blue threshold line should be pretty much overlap with the red threshold line. There is a problem. And also, you see the histogram. There is a big histogram around, almost around 6. So a lot of these are characterized with a problem. Okay, to solve this problem, clearly there is a problem with the thresholding. If we move up the absolute threshold up a little bit, and we probably will be able to mark those very bright areas as alveolar wall and being able to separate all the airspaces. Let's set this number and try again with a single test. Just test with this single image. Okay, yeah, while it's running the test, let's look at airspace profiling settings. Basically, most of the time, you should choose the automatic cutoff. So if you choose one distribution characterization, it's going to profile this based on size distribution alone and 95% upper confidence interval here. It's going to use this value to distinguish all the airspaces. On the left side, smaller than that will be colored green. On the right side will be colored red. Two distribution characterization will basically rely more on the red line, where you have two subpopulation from the area weighted size distribution, and take that cutoff in between these two subpopulations. Oh, this time the result is in. It's a perfect match. These two subpopulation has been matched almost perfectly with the Gaussian Gaussian distribution. And in this case, the blue and the red vertical line, they pretty much overlap with each other. Okay, and this time the cutoff is 2.8, 3.5. That's about right. Let's just allow it to color it. Before the color really come in, we can look at the profiling step, how each airspace was divided. Okay, look like this time it's much better. Those areas that's supposed to be separate from each other are really being separated. Okay, yeah, this is much better. And yeah, and this area used to be all dark because it's connected to the outside of the lung, and those count towards maximum size. Those count towards as part of outside of the lung. So those has been excluded in the previous run. Now, after setting a higher absolute threshold, it's clearly registered correctly. Okay, yeah. In a while, I guess we're gonna see the three-colored map here up refreshed here. Okay, before that shows up, okay, yeah, it's here. Okay, this view, 6 result view, there are a lot of information right here. If you click Start Batch, the batch processing will save this file, but even in a single run, you will be able to see all the results here. So this is where the file was found. Like the pixel scale was originally 1, and then you downsize this a little bit. Otherwise, it will be 1 divided by 1. Okay, the saccular threshold, ductal destructive threshold values, and the lung, based on your correct pixel scale, is going to calculate the size. It occupies 38% of the entire image. Below, which means whatever airspaces that are much smaller than this minimum size, all of those adding together didn't count as much. For those over, much larger, over the maximum size, they account for 62% of those. Whenever you preview an image, your exclude map will only show the green, but when you complete the test with minimum size and maximum size set, you see a lot more white areas. Those are excluded because they are bigger than the maximum size. Okay, let's switch back to the results. Okay, and in this case, parenchyma accounts for 88% of the entire lung, excluded 4%. This 4% is 4% out of the entire image. That's why this is kind of different. It's not like 4%, so this is 96. It's not. It's out of the lung. And next step, the septum, the alveolar structure, accounts for 38% of the parenchyma. Airspace, all the airspaces here accounts for 21% of the entire image. Whenever I didn't leave anything there, it's the entire image. And on the bottom, we can see all the size related parameters, or the count per area related, or the boundary per area, area per area, or also known as fraction. All of these values have been shown here. Destructive fraction of 35, close to 36. I think this is relatively healthy lung. Exactly like I marked. This is from air group. Didn't get a lot of smoke. Okay, overall I consider this well done. Profiling, categorization, and coloration of the entire image. A few more things I probably want to mention to you. Multi-threading. Sometimes it can speed things up, but the drawback of multi-threading is you will not be able to see picture-by-picture preview here. Whenever you do not check this on, when you finish one image, all the views will be refreshed, so you can get direct feedback on how well it does. Export full resolution. This is helpful if you do not like the resolution that you receive. When you turn on start batch processing, the final image probably have this much of a resolution, which is perfectly fine with me. But if you want more details, almost like 100% detail, you probably want to check this and start batch processing. That's pretty much it, I think. Alright, thank you for watching. If you have any questions, please leave a message. Let me know. I'll get back at you. Alright, thank you for watching.